Hey y'all, it's Dr. B. Little video on CpG Islands and methylation of DNA. To give credit, I'm using a video from Hussein Biology just to kind of talk over it. It's very well done. So again, we've learned about this before in class, but CpG Islands, basically cytosine, phosphate, guanine, dinucleotides, and there's usually a lot of them. They're generally found near promoter regions of genes. So basically understanding something, we're not talking about C's and G's where these are all in different strands of DNA. We're talking about cytosine, phosphate, guanine on the same strand, and you can see the P is for the phosphate, and therefore you're going to have kind of the, in essence, the same thing on the other strand, but obviously off one. So this is what we're talking about right now, is CpG repeated. So just because you have C's CpG somewhere, does that make it an island? The answer is no. You have C's next to G's all over the place. But one of the ways that we can identify them here is if you have more than 200 base pairs of CpG, PG, Cp all the way down the line on one strand, where the GC content is generally greater than 50% of them. And so that's how we identify these areas. And these areas are basically called islands. And in addition, about 60% of promoters in mammalian cells have CpG islands, and most of them are unmethylated, and that has a lot to do with their ability to be transcribed and translated. That's kind of the second part of this. The first one is identifying what a CpG island is. And the second part has to do with methylation. So the islands have a role in many things. Cell type expression of specific cell types, which makes sense. Again, think about the idea that, you know, we have all the same DNA in a cell of our finger. We have exactly the same DNA, in fact, in the cell of your finger as you do from a cell in your eyeball. But yet they're different cells. Why? Well, part of it has to do with suppressing and activating different genes to produce the different proteins. Also, suppression of some testis-specific genes has a lot to do with development and control of imprinted genes. And along that note, obviously, X chromosome inactivation makes a lot of sense. You can ask the calico cats about that one. So, really, what about them? Why do we care? Well, the answer is because of DNA methyltransferases, and these are the enzymes that do the methylation. And they love CpG islands, and we're going to see why. And finally, there's classes of the DNA methyltransferases. DNMT1 is a big player in the actual methylation itself, but 3A and 3B also have roles in maintenance as well. Just to give an example of one, how obviously we're talking about here, it's mainly the cytosine that is methylated, and basically how that happens here is you can see that, for example, in this case, DNMT 3A or 3B would catalyze through the addition of SAM a methylation at this particular location. And so therefore, you can see that this particular cytosine is methylated, and that, of course, is going to inhibit transcription. It's going to help to keep the histones very tightly packed, and therefore not give any access to any of the other major enzymes that would want to be starting the process of transcription, like transcription factors, etc. So that's just a generic overview of what a CpG island is, and hope you enjoyed.